It's a pleasure to be here. As Clarissa said, this is my second time in Brazil. The first time was about 15 years ago, and I had a chance to see Salvador in Bahia. But this is my first opportunity to see what, oh God, to see Rio. And I'm looking forward to spending a couple days and seeing your beautiful city. This is my beautiful city, except it's a little tiny city. It's Gainesville. This is the medical complex at the University of Florida. And my lab is here in the academic research building. I'm on the 4th floor, and Lucinda Sach here is hiding out on the 2nd floor, so we get to see each other often. What I'm going to do today is to tell you some of the science that we are doing in my lab, and tell you the story of the virus that we work on. It's a relative of the virus that Clarissa's lab and Nissan's lab works on, but it is different. And I'm going to tell you a little bit about the history of this virus and show you some of the projects that we're doing right now in the lab, and tell you a story about how some of those projects ended up being very different than when we started doing them. And I'm going to tell you, it's a curious road because the road that we are on was led by curiosity. I never meant to study cancer, but it turns out the virus I will describe to you has a natural ability to infect and kill human cancer cells. And I'll tell you about that story and how we came about. And I'll show you some data that led us to go down this road. And I hope you will find it as curious as I have found it. So, to begin with, like most directors, I say we a lot, and we means something, and this is what we means for me. These are the people in my lab. These are the students, the postdocs, the senior research associates, the people doing the real work in the lab. I get to go around and tell about it and write grants to get it, but these are the people who actually do the work. And I don't have time today to tell you about all their projects, and I only have really time to describe what one or two of them are doing. But I'm going to focus a lot on the work of Eric Barty, who's a postdoctoral fellow in my lab. He was a graduate student in Klaus Frew's lab before he came to my lab, so his history was studying herpes virus, and now he's studying Poxvirus. And I'll also talk a little bit about Masmuda Rahman, his work. Masmuda came to my lab when I was in Canada. He came from Bangladesh and knew a little bit of virology and has actually become an expert in poxviruses, and I rely upon him a lot to help me running all the different aspects of my lab. So to begin, I thought I would describe the field that I'm in to you. A couple years ago, Brett Finlay, who studies bacteria, and I were asked to write a review summarizing how bacteria and viruses inhibit the immune system and subvert the immune system. Amazingly, until we wrote this review, no one had ever done that. There are thousands of reviews about how viruses subvert the immune system, hundreds of reviews of how bacteria do it, but no one had ever written a review comparing them. So actually, Brett and I learned a lot in doing this review. And one of the things we learned is that pathogens of different kinds, bacteria and viruses, understand the immune system better than we do. And in fact, if you look at their targets, what pathogens have to subvert in order to survive, what we learn is that many pathways are in common. Many target pathways are in common. And so, for example, the NF-kappa B signaling pathway is targeted by bacteria, by other kinds of intracellular pathogens, bacteria. It's really remarkable. And so, as we were putting this together, we were looking for a term to describe what it is we were doing, what it is we were summarizing. And so we came up with the term anti-immunology. So all of you are familiar with immunology. It's the study of the functioning of the immune system. Well, anti-immunology is the study of how pathogens subvert the immune system. And it turns out the mechanisms and strategy of anti-immunology are every bit as complex as immunology, but both exist with respect to each other. One is a doppelganger of the other, if you like. And so my career, the past certainly 15 years of it, has been studying this particular aspect of viruses, namely how they subvert the immune system. And that study has taken us on many unexpected roads, and today I'll describe one of those roads to you. So I'll begin with Poxvirus. Those of you that know Nassim and Clarissa well know this already, but for those of you that don't, Poxviruses are large DNA viruses that affect a whole spectrum of eukaryotes, and there's also a family that infects insects. I'm just going to talk about the Poxvirus, which are those that infect hosts with a vertebrate backbone. Poxviruses are large, complicated viruses. The virion has almost 100 polypeptides, very complex, still never been crystallized. Fascinating virus family. And there are all kinds of poxviruses. They infect all kinds of vertebrate hosts. I'm going to tell you the story today of the poxvirus that infects rabbits. And I'll tell you why I ended up picking that system to study. A couple things that you probably should know to understand a few of the things I'll be saying in the talk today is that, number one, poxviruses are sort of unusual amongst DNA viruses. Most DNA viruses go to the nucleus, they enter the nucleus, and they parasitize elements of the nuclear machinery. Poxviruses are one exception to that. They replicate out here in the cytoplasm. They don't really need a nucleus in order to survive and then replicate in a mammalian cell. The other thing to bear in mind is that poxviruses bind mammalian cells in a very promiscuous fashion. The majority of mammalian cells will bind and allow the majority of poxviruses to begin infection. There are some exceptions to that, and I'm actually going to mention an exception later on. But in general terms, most mammalian cells bind most poxviruses. But what happens after binding and entry depends upon the individual poxvirus and the individual mammalian cell. In this case, I show a replication cycle leading to the eventual generation of progeny virus that goes on to infect new cells. This is called a permissive infection, and it simply means the mammalian cell has allowed the replication cycle of the virus to complete and create daughter viruses. That doesn't always happen. Sometimes the virus cannot go all the way around, and we call that a non-permissive infection or a restrictive infection. When that happens, it's usually because the cell has been able to beat back the virus, has been able to win the tug of war between virus and cell. When the virus makes it all the way around, the virus wins the tug of war. And we ended up studying the difference between a permissive and a non-permissive infection. In fact, we still study that in the lab because there are many important issues that we still have not learned what determines whether or not the replication cycle will be successful or not. And it turns, the answers that we have so far have turned out to be quite fascinating, I think. So the story that I'm telling you today actually begins here in Brazil, in the rainforest. And it began accidentally in Montevideo, Uruguay, when an Italian researcher by the name of Giuseppe Santarelli brought some rabbits to Montevideo. This was in 1896, more than 100 years ago. And the reason he brought some European rabbits over was that he was interested in using them as subjects in his tests in his laboratory. But he housed the rabbits outside, and very soon after he brought the rabbits here, they started getting sick, and they all died. And they all died of a horrible disease, and he knew nothing. He'd never seen this disease, didn't know anything about it. And he did inquiries, and no veterinarian had ever heard of it, and there was no record of it in the veterinary literature. So he figured it was a new disease, but he didn't know what it was. And he began conversations with microbiology colleagues, including a person by this name, Henrique de Beaurepaire Aragao, very famous Brazilian microbiologist. And Aragao began a quest to identify what this pathogen was that killed Santarelli's rabbits. It took him about 20 years. But his inquiry is actually an early example of forensic microbiology, very much like CSI for rabbit disease in this case. He uncovered what the disease agent was. It turned out it was a virus, and he isolated the virus, and he showed that the virus normally lived in the Brazilian rainforest in an animal called the tapete. It's also called the Brazilian brush rabbit. And in the tapete, this virus causes no disease. It's a very quiet infection, almost like CMV in people, very subclinical. But when a mosquito bites an animal, a tapete, having this virus, and then next bites a European rabbit, which is a member of a different genus, a different species, all of a sudden this virus caused a completely new disease that we call myxomatosis. And so he described this chain of events in this paper, and this was pulled out of the archives here in Rio by my friend and colleague Nissim Basach a number of years ago, because I couldn't get this paper in Canada or the US. So Nissim went to the library and found it and made a copy of it. And this is actually the first representation of a myxoma virus infected cell in the published literature. It's a beautiful paper. It's obviously written in Portuguese, so I can only read translated parts of it. But I can certainly understand this. This is actually beautiful representation of what a myxoma virus infected cell looks like. And in the cytoplasm, you can see viral factories, which is very characteristic of poxvirus infections. This comes from his 1927 paper, and this shows a rabbit that's in the process of dying from this disease. And Aragao deserves credit for finding out where this virus came from, identifying it, understanding what its family members are, and pointing out in the literature that this was a case of a virus leaping species from one animal where it had spent millions of years co-evolving into a new host. And when the virus got into the new host, it caused a completely new disease that no one had ever seen before. Viruses do this. HIV does it. HIV is not a human virus, as you all know. It's called human immunodeficiency virus. It's not a human virus. It's a monkey virus. And in its monkey host, it does not cause the immunodeficiency that we see in people. AIDS is a new disease of Homo sapiens. And this commonly happens in virology. And we have an example of that here, except the recipient host is not human. It happens to be Ornithologous cuniculus. So that's an important point to bear in mind. And when I teach this to students, I like to show this slide to remind them that a lot of people think bunnies are all the same. But that's like thinking primates are all the same. That's confusing you with an orangutan. So it's important to a virus knows the difference. And there's a very large genetic difference between a European rabbit and a tapete. They look similar, but genetically they're not only a different species, they're a different genera. So a different genus. And so when the virus is in this host, very quiet, in this host, absolutely lethal. Totally different. So this is a European rabbit. This is a feral European rabbit that happens to be in Australia. And one of the reasons that I'm showing you this is part of the story then after Aragao was finished. next popped up in Australia. And it happened to do with an accident in which rabbits like this were introduced into Australia about 150 years ago. And in Australia, these animals had no natural predators and lots to eat. And if any of you know rabbits, you know what they did. They made many, many more copies of themselves. In fact, they made 1 billion copies. And here's an example. This is in Australia of an area that was taken over by feral European rabbits that are not native to Australia, but now they're what Australians call an infestation. And you can see why. The rabbits have eaten everything and have destroyed the ecological environment. And so as a consequence, Australian farmers and the government declared war on the rabbit. This is about 100 years ago. And so they went looking for ways to kill rabbits. And eventually they found a way. They found a microbiological agent, a virus, that would infect rabbits, kill them, but nothing else. And so they imported myxoma virus, and after many years of study, released this virus into the rabbit population in southern Australia with the idea of creating a pandemic that would kill as many rabbits as possible. And so they brought in myxoma virus, and the lead PIs of that study were Frank Fenner, McFarlane Burnett, and Clooney's Ross. Frank Fenner was one of the great virologists in history. He just died last year. He was one of the leaders of the eradication of smallpox on the planet. McFarlane Burnett went on to win a Nobel Prize, and he's one of the fathers of immunology. Clooney's Ross was a senior director of the organization that allowed this virus to be released into the Australian subcontinent as a way to control rabbit populations. As part and parcel of their due diligence, that's what we call it, to make sure nothing bad will happen, Frank and McFarlane and Clooney's injected themselves with live virus to prove it was not a human pathogen. Sometimes we laugh about it, that's something Australians like to do. Because many years later, another Australian who studied Heliobacter, or studied the link between Heliobacter and ulcers, took the same tact of injecting, of basically drinking Heliobacter to prove it causes ulcers. He got ulcers. And he cured it with antimicrobial agents against Heliobacter. So Australians are good at doing this, but science has learned a lot from what they've done. So I'm not going to tell this story today. If any of you are interested, Frank Fenner wrote a book called Myxomatosis, in which he showed what happened after live virus was released into the feral European rabbit population. And this comes from one of his reviews. This area in black that you see here is an area that was almost completely sterilized of rabbits within about 12 to 16 months after the release of the live infectious virus. This was in 1950, 1951. By about 1953-54, the Australians thought they had cured the rabbit population using this new pathogen that was introduced by man into that population. But Mother Nature didn't agree. What she did is that approximately 0.1% of all the rabbits who were infected survived. And they were an allelic subpopulation that was naturally more resistant to the killing effects of this virus. So many of you that are familiar with virology know what happened next. The 0.1% of surviving rabbits got together, found they had lots to eat, no natural predators, and you know what they did? They made more copies of themselves, and the bunnies are now back. But now the virus is enzootic in the population. And the virus and the host are co-evolving together into a new climax association in which fewer of the animals died, and the virus itself is less pathogenic. It is still replicating and transmits well by mosquitoes, but it kills fewer and fewer of the hosts. So you can see a kind of co-evolution happening in real time as we watch it, of the evolution of a vertebrate host being selected by an extreme pathogen of that host. And this is one of the few examples where we can actually watch it happen in real time and study the consequences. So at any rate, myxoma virus, there's two hosts. There's a long-term evolutionary host where in the Brazilian rainforest this is not a pathogen. But when that virus gets out of the tapetti and into the European rabbit, now it causes a new and lethal disease that we call today myxomatosis. And this is such a deadly disease if you're a European rabbit. The LD100, that is to say the lethal dose it takes to kill 100%, essentially 100% of host animals, is a single infectious unit. Which means that as soon as any cell anywhere on a rabbit is infected with this virus, that animal will die with 99.9% certainty within about 2 weeks of the infection. During that infection, there's a complete collapse of the cellular, all aspects of the immune system of the host animal, and the virus replicates like crazy in all kinds of internal tissues. Here's an example. This is a rabbit skin section, and the stain that you see here, the brown, is just staining for viral antigen. Actually, in this case, it's beta-galactosidase that's been engineered into the virus, but it doesn't matter. The point is, where you see brown stain is where the virus is. And it just replicates everywhere. It replicates in fibroblasts, endothelial cells, myeloid cells, all kinds of cells. It's really incredibly promiscuous, and the end result is that this animal is doomed. So I actually have a movie here showing what this virus does in tissue culture. So in this movie, we've taken virus and we've engineered it to express EGFP. This construct expresses EGFP, enhanced green fluorescent protein, so that when cells turn green, that's an evidence that they have been infected and they're making gene products at the same time as they're making all the viral gene products. So this is a 27-hour movie. And so what you can see is green appearing here and there. These are cells that have been productively infected with the virus. And now you'll see them spreading to neighboring cells because they spread from cell to cell. And eventually, if we let the movie go long enough, all the cells would turn green. And eventually all these cells will die. But unlike some viruses that are very cytotoxic and very cytopathic, it doesn't create a hole in the monolayer. It creates lumps of cells, and we call these foci. So we titer myxoma virus based on its ability to cause foci in culture in the lab. And we count these, and that's how we titer the virus for all of our experiments. Now, one thing that you'll note here is that this is what the virus does in rabbit cells, and I've already shown you what it does in rabbits. But in the laboratory, these aren't rabbit cells, these are monkey cells. And the virus grows very, very well in monkey cells, and actually we grow up our large-scale preparations of the virus in monkey cells. And yet, if you take this virus and inject it into any non-human primate, it vanishes, it doesn't replicate. So I'd like you to hold that thought, because I'm going to come back to it. But it points out a very important aspect of viruses and tropism and disease, which is, why are viruses different in the laboratory and culture compared to an animal? And I'm going to return to that point. So many years ago, we realized in order to understand this virus, we needed to know the genes, so we had to sequence it. The genome is 160 kilobases, and it holds 159 unique genes. And they're just shown in different colors here. The colors are meant to designate the class of gene that it is. And the genes that are in red and yellow are genes that are very similar from one poxvirus to another. So if you were to pick a red gene, you would find a close homologue, or actually what's called an ortholog, in vaccinia virus or any of the poxviruses that you study in this institute. On the other hand, the genes that are in green here are host interactive. and they are designed for the virus to survive in a vertebrate host. So generally, if you take a green gene and knock it out by using recombinant DNA technologies, the resulting virus is live, it grows in culture, but it no longer causes the same disease in the infected host because it's been crippled by losing some of its anti-immunology repertoire. The genes that are in black, we don't have a clue what they are. The sequences don't tell us, and we just have no information. Quite likely they're host interactive or immunomodulatory. It just, at this point, we don't know. Sometimes when a student or a postdoc comes into my lab, if they're bold, they'll take a black gene and study it. And their job is to turn it into a green gene. And if they succeed, they write a paper and everyone's happy. So over the years, we've knocked out many genes. Generally, we focus on these genes that allow the resulting virus to survive. But they affect the biology of the ability of the virus to cause disease. And over the years, we've knocked out different genes and we've studied them, and I've learned a lot. In many ways, everything I've learned about immunology has come from studying knockouts like this and examining the effects of those knockouts. And almost always when we knock out a gene, we learn something new about the biology of the virus or about the disease process. And sometimes we are surprised and sometimes we are astonished by the properties of an individual knockout. When I go to Poxvirus conferences, people in my lab take one or two genes and describe the results of a knockout and describe what we've learned. So for this talk, I'm not going to do that, but I just will mention one or two examples to give you a feeling for how sophisticated and knowledgeable these viruses are compared to us. Here was an example in which we knocked out a very tiny gene near the ends of the viral DNA called M13. M13, which is shown here, is a single domain. It's a tiny little protein, and the only thing database analysis tells us is that it has the motifs of a domain called a pyrin domain. And those of you that study immunology, or those of you that are familiar with inflammasomes, which are an organelle that help control innate pathways in myeloid cells, will recognize that an inflammasome is a complex that is found particularly in myeloid cells that controls the activation of a number of cytokines, pro-inflammatory cytokines, particularly IL-1 beta and IL-18. And it does it by controlling the activation of an enzyme called caspase 1. It used to be called ICE, now it's called caspase 1. And it is the complex, the inflammasome complex, that responds to upstream pro-inflammatory signals that when it turns on, it activates caspase that then drives the secretion of these cytokines. So it's like a gatekeeper for inflammatory responses from myeloid cells. What this viral protein does is it goes in and insinuates itself into the complex by binding to a molecule called ASC1. When it does that, it turns the caspase, the inflammasome complex off, and it no longer responds to these upstream inflammatory signals. So it's a very sophisticated strategy by the virus using a very small protein to turn off a very complex cellular organelle. Very clever, very sneaky, very surgical. It's really amazing. So one of the things that we did is not only did we knock this gene product out, but we expressed the protein in using recombinant technologies and asked, what else does this protein bind to? And this is what we call classic proteomic fishing analysis, using the viral protein as the hook, and we see what fish do we pull in. Well, not only do we pull in ASC1, but when actually we did those experiments, we pulled in another fish that we never imagined, and it's a molecule of the NF-kappa B signaling pathway called NF-kappa B1. It's actually called P105, which is a precursor molecule that normally has to be cleaved when the NF-kappa B signaling pathway is turned on. What the viral molecule does is it binds P105 and it stops its degradation. It stabilizes P105. And by doing so, it gums up the NF-kappa B signaling pathway by a new mechanism. And it's not a pyrin-pyrin interaction domain mechanism, it's another mechanism, and we actually don't understand it right now. We've written a grant to try and be given permission to tear apart the mechanism, but right now we don't know what it is. All we can tell you is P105 does not have a pyrin domain. So it's not obvious, just looking at it, why that viral protein is an inhibitor of that pathway. But it is, and it's a beautiful inhibitor, and it's an example of yet another activity of a relatively small viral protein that attacks another pathway important for immune responses. Just to leave you with one thought before I actually take you to the story, poxviruses and many viruses are good at making secreted proteins, not only intracellular proteins, but secreted proteins as well, that have different activities against the immune system. And the reason I wanted to mention it is that one of these, it's a virus-encoded molecule. It's a serine proteinase inhibitor called SERP1. So myxoma virus has a number of inhibitors of the serpin superfamily. One of them is secreted, and it's called SERP1. The reason I wanted to mention that is that SERP1 has been used as a drug in human clinical trials to treat diseases caused by excessive inflammatory activity. The clinical trial that it's been used for is for acute cardiovascular syndrome. Basically, patients that come into the emergency with heart attack. Many of them are systemically inflamed where their myeloid cells are turned on and they can't be turned off. Nobody knows how to turn them off. It turns out this viral protein knows how to turn them off because that's what it's been doing for millions of years for the virus. And so that study was just reported last year. It was run by Alex Lucas, my wife. She's a cardiologist who's actually taken some of these molecules and shown in clinical trial that they can inhibit their target in the absence of virus. And so we believe that viruses are a new source of pharmacological drugs to treat all kinds of diseases that have nothing to do with viruses, but everything to do with immunology and inflammation. So this is the very first study of someone has used a viral protein as a therapeutic. And one of the reasons that Alex actually isn't here with me in Rio is she was just presenting this data yesterday to the World Serpin Congress, which is in North Carolina. So I'm not going to talk about this anymore, but it just as a thought I can put to you is that not only do viruses cause disease, but they can be a source, a remarkable source of pharmacological reagents for things that have nothing to do with viruses, but everything to do with either inflammation or immune pathways, because the virus is very good at making inhibitors of those pathways. But that's not what I'm going to talk about today. Instead, I'm going to tell you about some experiments that we did that ended up taking us on this curious road that I told you about to begin with. And the story starts with a very simple question. And it's a question that virologists ask themselves all the time. And it's easy to ask, but harder to answer. And the question is this. When you look at viruses in biology, particularly viruses that cause disease, the one thing you notice about the virus is they're very picky. They're very specialized. They don't cause disease in all kinds of hosts. They tend to be very specific. The famous example of that is smallpox virus. It causes disease only in humans, no other animal. It's a pox virus. It looks a lot like myxoma virus. If you look at the particles, very similar. The genomes are very similar. But one is rabbit-specific and this one is human-specific. And yet we don't know why. We look at the genes and we do all the database analysis we want. We cannot look at it and say, that's a human virus, or that's a mouse virus, or that's a camel virus. Biology knows, Mother Nature knows, and we do if we do an experiment like this, but we don't understand it. And it begs a simple question. Why are pox viruses in this case, but you can say it for any virus as well, why do they tend to be so species-specific in their host tropism when many of these viruses in the laboratory in cultured cells often do not show that specificity? So all of these viruses you can grow in monkey cells, for example, but if you inject that any of those viruses into monkeys, it vanishes. Even smallpox is not really a monkey pathogen. So what got us into this were some experiments we were doing a number of years ago when my lab was in Canada that ended up giving us some results we did not anticipate. And here's an example of it. These are mouse embryo fibroblasts. So these are primary fibroblasts just taken out of a mouse, grown in culture. And we infect them with myxoma virus that expresses beta-galactosidase. And what we found is a couple cells would turn blue, but basically the virus would vanish. It would just completely go away. And after a while, you couldn't find live virus anymore, but the cells were there and they were happy as clams. So this is a classic non-permissive infection, and the virus could not make it through its replication cycle, could not make progeny virus. And for years, we did not understand this phenomenon. If you had asked me 5-10 years ago what happened here, I would have said, maybe there's a defect in the receptors of these cells, and that's, in part, it was based on ignorance because we didn't have a clue what the answer was. And that was the best guess I could have given you. But a person in my lab began doing some experiments that changed that view very dramatically. Fuan Wang, a senior researcher in my lab, was fiddling with a signaling pathway called the interferon response pathway. And what he found is if he took these cells and ablated interferon signaling, either by using neutralizing antibody to delete the ligand, or looking at cells that had a genetic deficiency in the receptor for type 1 interferon, or using cells that were deficient in signaling for interferon. It didn't matter. The end result is we went from a non-permissive phenotype to a fully permissive phenotype. When you think about it, that's pretty amazing. We've gone from a mouse-like phenotype to a rabbit-like phenotype in the same cells simply by interrupting one signal transduction cascade, the type 1 interferon signaling pathway. That's amazing. And it got us to thinking, well, maybe tropism wasn't so much a receptor phenomenon, maybe it was a signaling phenomenon and had to do with the ability of the virus to either turn on or turn off signaling pathways of the infected cell. So this then led us into a number of inquiries. I'm going to show you some of the data that actually I didn't show at the Congress I was just at in Achibaya, and show you some of the data that led us on to this oncolytic virotherapy pathway. So another thing we noticed as we were doing these experiments is that if we took primary murine embryo fibroblasts, infected them with virus, they were non-permissive. But if we took the same cells, put them in tissue culture, and kept passaging them until they immortalized, spontaneously immortalized, and most cells do this. And if any of you are working with mouse embryo fibroblasts, they're probably immortalized, unless you isolated them yourself. When we did that, we found the phenotype of the cells transformed from this phenotype to a permissive phenotype. Again, this was amazing, and we couldn't explain it. But at this point, we knew that this phenomenon had something to do with the interferon pathway, but we didn't know exactly what aspect of the pathway. All we could say is when we infected these cells, they were non-permissive. We infected these cells after immortalization, they reverted phenotype into the permissive phenotype. And so, to make a long story short, we were able to show that what happened is that these immortalized cells, and as a property of their immortalization, lost the ability to sense the virus infection, and they started producing type 1 interferon. And that turned the virus off. So these cells sensed the virus, did not sense the virus properly, did not turn on interferon. Let me back up. These cells are non-permissive because they induced interferon. They sensed the virus and produced interferon. These guys lost the ability to sense the virus, turned on interferon, and that made the cells non-permissive. That's what I meant to say. So that was telling us the ability to sense the virus is intrinsically entwined with the tropism. So then we thought, what's going on in human cells? So we got human fibroblasts. And what we found, primary human fibroblasts did not behave like primary mouse fibroblasts. Instead, they behaved like transformed mouse fibroblasts. Even though these are new, fresh fibroblasts, they're fully permissive for the viral infection. This was another amazing result, completely contrary to what we expected. If you'd asked me before we did the experiment, I would have said these would be non-permissive. But they are. They're fully permissive. And the reason is they don't induce interferon when they're infected with the virus, the way primary murine cells do. And again, this was pretty mysterious because if we take this virus and put it into people, it vanishes. So something is killing the virus in people. And again, to make a long story short, Eric Barty in my lab figured that out. It turns out there were 2 cytokines you need to wipe out this virus in human cells. One is interferon. The other is another cytokine called TNF. And when you add interferon and TNF together on primary human cells, it wipes out the virus. Okay, so now it's starting to make some sense. But something must be making the interferon and TNF. Something must sense the virus in people and make interferon and TNF. So we figured if it's not fibroblasts, it must be another kind of cell. So we screened other kinds of primary cells until we found out that primary human macrophage sense this virus, turn on TNF and interferon, and that shuts off the replication in macrophage, in myeloid cells. So now the picture is getting a little bit clearer. If we take macrophage and fibroblasts and mix them and throw virus onto that population, that mixed population, guess what happens? Anyone? Any of the students? What do you think? You mix macrophage and fibroblasts, mix them up, add virus, what do you think happens? You have to say it in English, sorry. I don't understand. Right, absolutely. So you mix the 2 cells, and all the cells are now non-permissive because the interferon and TNF made by the macrophage protects every single cell in the population. Makes sense. Now we're beginning to learn why it's not a pathogen in real people. There are cells that turn on interferon and TNF, and that shuts the virus off completely. Okay. So in data I won't show you, what that said is that macrophage have a sensor that recognizes this virus. And so we went looking for the sensor, and we eventually identified the main sensor. It's a molecule called RIG-I. Very famous. It's well known for sensing RNA viral infections. But this was the first discovery that RIG-I could sense DNA virus infections, because the ligand for RIG-I is RNA, not DNA. So at this point, we're not sure what aspect of the double-stranded DNA or 5' triphosphate RNA is the actual ligand for RIG-I. It doesn't really matter for the point of the story. The point is that myxoma virus signals through RIG-I and turns on interferon and TNF in myeloid cells, and that shuts off the replication of this virus in any primary human cell. It doesn't matter what lineage it is. Interferon and TNF kill it. So one of the things we wondered is, so what is interferon and TNF doing in mammalian cells, in human cells in particular? So we did this with microarray analysis in conjunction with the chair of our department, Henry Baker, and this is what we found. So these are human fibroblasts, primary human fibroblasts, in which we've thrown on either TNF or interferon or TNF plus interferon. And you don't have to be a microarray expert to realize that when primary human cells see TNF, they turn on a bunch of genes. Actually, there's about 300 genes. When interferon infects, they turn on about another 300 genes. A different set of genes, but it's an interferon response pathway. But look what happens in human cells when you add both cytokines at the same time. No one had ever done the experiment. Very simple experiment. Hundreds more genes are turned on. This is a new antiviral state. At least in terms of, in nature it's not new, but in terms of our appreciation of it, it's a new antiviral state. So it involves the induction of hundreds of genes that are not turned on by TNF alone or interferon alone. These are new antiviral genes turned on, and you need both cytokines in order to turn them on in human cells. So one of the things we did is we made RT-PCR primers for these new, what we call synergistic genes, and we went looking in different kinds of cells for cells that are capable of synergy, that is, capable of seeing interferon and TNF and inducing these genes the way most primary cells do. And what we found is the list that we published elsewhere, so you don't have to go into it. But these are a new set of genes turned on, but you need both cytokines to turn those genes on. The amazing thing we stumbled upon is when we went looking for these responses in cancer cells, we couldn't find them. We can't find a single human cancer cell that's capable of a normal synergistic response. They're all defective genetically in this pathway. So we wondered whether or not this would be one of the explanations for why this virus infects human cancer cells so well. Joanna Saipula, a graduate student in my lab, before actually we did these experiments, screened a whole variety of human cancer cells. And what she found is something like 2/3 of all human cancer cells behave like rabbit cells in the laboratory. So it was really this observation that got us into the field of oncolytic virotherapy, which simply means using live viruses to therapeutically treat cancer for the purpose of infecting and killing cancer cells, but leaving normal cells alone. So it's different than gene therapy, different than vaccination strategies, but it is a novel mechanism, if you like, for attacking cancer using live virus. The truth is, oncolytic virotherapy as a discipline has been around for 100 years. And there are books and reviews written about the history of oncolytic virotherapy. But it's only more recently that with the technologies of modern recombinant DNA strategies that people have been able to look more carefully at live viruses to treat cancer. And so there's a field of oncolytic virotherapy out there. We meet every 2 years. The last meeting was in Arizona. And when we get together, we sort of compare notes. What we are all looking for is a virus that has these properties. It has to have low pathogenicity. Obviously, it does no good to wipe out the cancer if you kill the host. That's not therapy. It has to have a high oncolytic capacity. In other words, the virus has to be able to distinguish a normal cell from a cancer cell somehow. It has to be able to replicate robustly and spread in complicated tissues, because real cancer is not like cells in a monolayer. They're full of different lineages of cells, and the virus has to percolate through them. You have to have a delivery method, and you may or may not want an immune profile. This is a very interesting and contentious issue, and I'm not going to talk about it actually today. We'd spend the whole lecture talking about it otherwise. But right now, the oncolytic viruses that are out there that are being tested, either in the laboratory or in human clinical trials, come in two types. There's human viruses and there's zoonotic viruses. The human viruses are viruses that are normally associated with people, but are generally not super pathogenic. And they include mutants of adenovirus, mutants of herpes virus, mutants of measles virus, sometimes the vaccine strains. For example, the vaccine strain of measles virus is in human clinical trial right now. Among the zoonotic viruses, there's only one other Poxvirus being tested in clinical trial for cancer, and that is vaccinia virus. It's actually a derivative of a vaccine strain called YF, which is being used in trial right now for liver cancer. And that trial is being conducted by a company called Generex, which is named after Jenner, who initially started vaccination or taught the world about vaccination with vaccinia virus. Myxoma virus is the newest kid on the block. We are still preclinical, but we're in discussions with biotech industry about making clinical grade virus and going into human clinical trial. And I'm going to end up by showing you what I think our clinical trial is going to look like based on what we've learned about how the virus operates. So we started doing oncolytic virus experiments about 5 or 6 years ago, And we started in collaboration with a neuro-oncologist, a guy named Peter Forsyth. He studied glioma, glioblastomas, different brain cancers. And he also runs a lab where he's looking for new therapies for brain cancer. And as Peter will tell you, and any neuro-oncologist will tell you, many of those diseases are bad news. Right now, medical therapy cannot save the vast majority of patients with glioblastoma, for example. The standard therapies are not good enough. And so many people are looking for new generations of therapies to work against brain cancer. So Peter had heard about our work and said, why don't we team up? And we'll use your magic virus tagged with EGFP, and we'll put it into a mouse bearing gliomas. In this case, the mice are bearing human gliomas, so the mice are immunodeficient to allow the xenograft to not be rejected. Normally, when you inject human gliomas into the brains of nude mice, this is what happens. They all die within a couple weeks. And that couple weeks is a window of time where you can do whatever therapy you want, add whatever drug, irradiate them any way you like, give them whatever therapeutic regime you want, try to save those mice from those gliomas. So here are some sections in which Peter's lab injected GFP-expressing myxoma stereotactically directly into the tumors. And what we found is that the virus replicated in the tumor just like it does in rabbit tissues. When it hit normal tissue, it stopped. And the tumors regressed until they all vanished. We can no longer even find them on sections. And when the tumor cells vanished, the virus vanished. So we converted a kill curve like this to a kill curve like that. Peter was very excited. I was very interested because how the virus behaved in tumor tissue looked a lot the way it behaved in rabbit tissue. And so that led to a whole series of collaborations with different groups. We've done some ourselves, some we've done with collaborators. The kinds of studies we've done in animals are two kinds. One are studying human cancers xenografted into mice or rats. The other is in murine cancers in either mice or rats. These have the advantage of studying human cancers. The disadvantage is that there's no immune system. These have the advantage of being in the presence of immune system, but the disadvantage is they're not human cancer, they're mouse cancers. So the FDA likes to see results in both pathways, and you can go into the literature. I can share you any of the papers. All of this has been published. I just want to end up with a story that is mostly unpublished now, that we think is informing a clinical trial that we think will be the first use of this virus in human patients. And it's a little different than what I just described to you. What it involves is a modification of autologous bone stem cell transplantation for cancer. Around the world, many standard cancer therapies either succeed or fail. When they fail, there sometimes is a backup strategy which involves using exceedingly high dose of chemotherapy, basically lethal chemotherapy, in the hope of wiping out every last cancer cell in that patient. The downside of this chemo, it's called myeloablative chemotherapy, is that it also wipes out the immune system. The drugs are so high, they're so toxic, maybe you can kill the cancer cells, but you also wipe out the immune system. So the antidote for high-dose chemo, myeloablative therapy, is stem cells. And stem cells, if it's done properly, can go in and reconstitute the immune system. It reboots the immune system in those patients. One of the problems of that method is you can get stem cells from the patient themselves before they undergo their high-dose chemo. And you can get it from their bone marrow, or you can get it from their mobilized peripheral blood. Both are used clinically. But one of the problems of getting it from the patient is sometimes those grafts not only have stem cells, but they're contaminated with the cancer cells from the patient. So when you re-engraft the cells, you not only reboot their immune system, but you reseed the same cancer back in those patients. So people have looked for ways to delete cancer cells from these grafts. It's called purging. And over the years, heme-oncologists have eventually given up on purging because no one has found a strategic way of getting rid of the cancer cells clinically They can do it in the laboratory, but clinically it doesn't work well enough. And so the field has almost given up on the idea of purging cancer cells from autologous grafts. So when I went to the University of Florida, I had new colleagues, including Ed Scott and Chris Coggle. Ed Scott is a stem cell transplant expert. Chris Coggle is a hem-onc person who sees patients with leukemia and lymphoma. And many of his patients get bone marrow transplants or stem cell transplants. So we wondered, maybe, you know, science, as we practice it now, isn't smart enough to know how to delete the cancer cells, but maybe viruses are smart enough. So we wondered whether or not our bunny virus could be used to delete specifically cancer cells from human bone marrow grafts. Now, for those of you that are familiar with stem cell transplantation will know this, but just for those of you that don't, there's two kinds of grafts you can give a patient. One is their own stem cells, and we call that autologous. And the other is a donor, usually an MHC-matched donor of some kind, and this is called an allogeneic graft. These grafts are the safest of all because it's self being transplanted back to self. The problem is, if they're contaminated with cancer cells, then you're reintroducing the cancer. These grafts can be very effective, but there's a disease called graft versus host disease in which the graft actually attacks the recipient patient. And even today, with the best modern clinical practice, something like 10 to 15% of all allogeneic graft recipients die from graft versus host disease. Big problem. Hasn't been solved yet. So I'm just going to talk about using the virus for deleting cancer cells from these autologous grafts. And I'll just quickly go through some data and then I'll finish. So different kinds of cancers tend to get different kinds of grafts, and clinicians have learned this over the years. Some get allogeneic, some get autologous. But amongst all of these, the disease we kind of focused on was multiple myeloma because so many of them get autologous grafts. So we thought this would be a good model system for us to ask, can the virus selectively recognize and kill multiple myeloma cells? So multiple myeloma is the story I'll tell you, and then we'll finish. So multiple myeloma is a very dangerous cancer. It's a cancer of B lymphocytes. The consequence of the transformation of those cells is they make copious quantities of plasma Ig, different kinds of IgG, depending upon the cancer. It amounts to tens of thousands of cases in North America and around the world, a huge number. It is essentially incurable. Modern clinical practice can buy patients time, but it can't cure them. My brother-in-law, my wife's elder brother, got multiple myeloma a couple years ago, and he had a genetic form that was resistant to current chemotherapy, and he lasted less than a year, and it's a horrible way to go. We regard multiple myeloma as essentially incurable right now. Although bone marrow transplants will buy patients time, often several years of good time before the disease relapses. So the first thing we wanted to know is, can this virus kill human, can it infect them, can it kill them? Human multiple myeloma cells. So we got a bunch of different human multiple myeloma cell lines. This is the work of Eric Barty in my lab, and just simply added virus to them and asked what happened. If we don't add virus, they grow like crazy. If we add virus, they just drop like stones. They're all killed in culture. That's good. So in culture, it infects them. If we take multiple myeloma cells and we add them into a normal bone marrow mixture and add virus to them in culture, we can detect the multiple myeloma cells because by FACS analysis, they've got specific cell surface markers that we can use. Here are the multiple myeloma cells we've added into this bone marrow culture. If we just simply add virus, that quadrant goes away. The virus infects them and kills them, even in the presence of normal human bone marrow. So that's good. But again, it's only in vitro. We next asked, did it do anything to the stem cells? Well, there's a couple different assays you can do to ask whether or not the stem cells are operational and functional or not. One is an in vitro assay called a colony-forming assay. And basically, you take human stem cells and you culture them in vitro, and you look for the generation of different lineages of immune cells from those stem cells. It's done all over the world, very common. It's called a CFC assay, or colony-forming cell assay. So if we do that with human bone marrow, we get all these different lineages of cells in these different proportions. And this is very normal. This is the result of a normal stem cell assay with human bone marrow. If we treat those cells with myxoma virus, GFP, and then do the same assay, it looks identical. So what this is saying is that in vitro, the virus doesn't hurt the stem cells that are in that population. We now know that, well, I don't know if I have the data or not. Maybe I'll come back to it and say we now know why. I'll see if I've got the data slide here or not. But that's all in vitro. What the FDA wants to know and what the field wants to know is, what about in vivo? How good are your stem cells in a real transplant scenario where you're trying to reboot an immune system? So short of doing that in patients, and we can't do that yet, the surrogate is to use highly immunodeficient mice. In this case, these mice are what we call NSG. They're NOD-SCID, they're the IL-2 receptor gamma chain. And these mice have no T cells, B cells, or NK cells. And so they receive human grafts very, very efficiently. So we can take an NSG mouse, sublethally irradiate it to clear out its own bone marrow, and then engraft in human cells of different kinds. We can use bone marrow, we can use cancer cells, we can use mixtures, we can use patient samples, whatever you want to do. And you can either treat it with virus or not, depending on what your experiment is. But the end result is that you cook the mouse, and then 2 months later you pull out its bone marrow and use FACS analysis to ask, are there human cells in the bone marrow, and are they normal or cancers? And we can do that with FACS analysis. Here's an engrafted mouse, here are the mouse cells down here, here are the human cells up here. So this is the experimental strategy for asking whether or not the engraftment potential of the cells that you start with is competent or not, or whether you've interfered with it. So if we do this with normal human bone marrow, what we see, here's the engraftment levels in the mock-treated controls. Here's the engraftment level in the mice when we treat the samples with virus first. There's no difference. We can't tell them apart. So what that's saying is that the virus treatment is, as best as we can measure it in mice, harmless for the engraftment of the stem cells in that bone marrow transplant sample. And here's the slide I was referring to. The reason it does that, we now know, and we use labeled tagged virus to do this, is that the virus, the virion itself, does not even bind the population of cells that contain the stem cells, which are CD34 positive stem cells in the human population. So the virus, if we do binding analysis, the red you see here is a positive control. The thing that's in green is a negative control. And the human CD34s that we can use FACS to gate on them simply don't bind tagged virus. That's why it's safe for human stem cells. It doesn't bind them. It doesn't even start the infectious cycle. So this is one of the few exceptions to the rule I told you at the beginning. The human stem cells are non-binders for this virus. So what about multiple myeloma cells? We do the same experiment. We put in the different classes of multiple myeloma cells. Here are the controls that you see here, the mocks, the virus-treated controls. We can't find a single cell engrafted into any mouse. It has completely purged the multiple myeloma from these samples. And the last piece of data I'll show you, because I know some of you have to go, what happens if we mix them? Mix normal bone marrow with multiple myeloma cells, mix them, add virus or not, engraft them into mice, wait 2 months. What we find is if we look for, these are multiple myeloma cells. In the mock-treated, there's lots of myeloma cells in the bone marrow. The virus-treated, no more multiple myeloma. It completely destroyed their ability to engraft. But the normal cells over here that we can gate separately by FACS analysis, the mock and the myxoma-treated, identical. It didn't hurt the engraftment of the normal cells in that population. It selectively deleted the myeloma cells, left the normal cells alone. That's where we are. That's the bottom line. So what I can tell you is that this virus doesn't hurt normal stem cells by every criteria, colony-forming assays or engraftment into mice. It eliminates a whole spectrum of leukemic or myeloma cells, and I didn't talk about the other cancers. I just talked about myeloma today. And so we are hoping is that we will be able to prove to the FDA and to the scientific community that we can use this strategy to not only make autologous bone marrow transplantation safer for patients, but maybe if we're lucky, we can make more patients eligible for ABMT by having a strategy where we can take their bone marrow, delete it of cancer cells using a virus, and then make them eligible for ABMT and rescue therapy. So that's our dream. I don't know if it'll come true or not. There's a lot we still have to do. We have to go to the FDA. We have to make clinical grade virus. We have to do a clinical trial. All that's in front of us. But, you know, wish us luck, because at this point, I'm hopeful, and we're in the business of being hopeful. That's why we do research, is because we have hope it will matter. And we will see in the future how it plays out. So that's where it is right today in 2011. So here's my gang. Some of the people have gone on to get real jobs. Here are some of my collaborators. I was funded by many agencies in Canada, and I'm really grateful to them. Now that I'm in the U.S., NIAID and NCI fund my lab. There's an agency in Florida called Bankhead Coley that uses money to try and promote ideas in the lab for clinical translation for cancer. And I want to thank all of those agencies. And I want to thank all of you for hanging around and listening to me in English. So thank you. If there are any questions, I'm happy to talk about them. Students are allowed to ask. Okay, go ahead.
That was a great talk.
Thank you.
Do you know what the virus binds to that's missing in the CD34?
We know something. In fact, we have a project that has just started to try and identify what the binding determinants are. What I can say is that we can block the binding in all cells with heparin. So we think GAGs, surface GAGs, are a key component. But we know that CD34s also have GAGs. So it's going to take us a while to deconstruct it. But we have one cell line where the virus does not bind, but when we add PMA to it, the virus starts binding. So at least we've got an experimental strategy. We can ask, what is PMA inducing on those cells, and can we deconstruct what the binding determinants are? I'm going to guess they're GAGs, but that's a guess at this point. Yeah?
How does a virus spread from one cell to the other?
It's by direct contact. So cells have to bump into each other, and then the virus actually gets passed from cell to cell. Not very much extracellular virions exist unless the cell disintegrates and the virus actually pops out. So we know a lot more about how vaccinia transmits from cell to cell than we do about this virus. It's just we haven't studied it intensively. But the observation is that at least at some point the cells have to contact each other. In an animal, it's spread by infected lymphocytes and leukocytes of different classes. So we know in the animal that's how it spreads.
Sorry.
Yeah.
I have a question about the sensing of the virus.
Yes.
You mentioned the
Yes.
Greek protein. Is it the only sensor in the cell, because there are some other?
Probably not, because what we found, for example, RIG-I is a predominant sensor in human macrophage. And yet, when we go to other cells of different lineages, RIG-I doesn't seem to be the sensor. So even if we take human myeloid cell lines, for example, in other words, transformed derivatives of those macrophage, RIG-I is no longer the sensor for the virus. So what it's telling me is that viral sensing in mammalian cells is very lineage specific, and it might even be specific on factors that we don't appreciate right now. So sometimes people ask me, what's the sensor? And I'm reluctant to say it's RIG-I, because it's only RIG-I in human macrophage in response to the wild-type virus. And the situation is probably more complex than that in other lineages.
So it hasn't been done in RIG-I knockout mice?
No, because we've actually moved almost completely to human cells. And so we tend to do knockdowns rather than knockouts, because we really want to know human lineages. And there are limitations to doing knockdowns in primary cells. We can get reasonably good knockdown of RIG-I using lentivirus, for example, expressing shRNA against RIG-I. But it's not as clean as knockouts in mice. But still, we want to know the answer in human cells, and we've seen enough to know mice lie. So we want to get our data in as best we can in human cells. So if someone will create a knockout RIG-I human, let me know. I want the cells. Any other questions? Yeah.
Have you thought of a strategy to try to move up to the clinic in cases like metastatic melanoma? How would you...
Right. Yeah, see, the question he's asking, what about other kinds of cancers like melanoma? In fact, we've actually tested melanoma in mice using the B16 melanoma model. And we have a clone of B16 that likes to metastasize to the lung. So we've actually done experiments on the B16 F10 clone, the lung metastatic variant in mice. And what we found is that the virus works pretty darn well, but we don't have a delivery mechanism to get the virus to every last metastatic site. And with cancer metastasis, being 90% efficient or 99% efficient or even 99.9% efficient isn't good enough. So we have not cracked the problem of delivery. How do you deliver the virus to every last cell? And that is an issue for oncolytic virotherapy in general. The oncolytic viruses that are in clinical trial, and there's one that's been approved in China right now. There's an adenovirus derivative that's been licensed in China for head and neck cancer. The problem with all of those is getting enough of the virus to every last cancer cell. It's a technological and a technical problem. It's going to be solved. Someone is going to solve that problem, but it's not solved yet. And so the reason we're heading towards a clinical trial with ex vivo therapy is I can get the virus to every single cancer cell because of the strategy. We circumvent that problem by using this as our model for the clinical trial. That's really the reason right now. If we get into clinical practice and learn how to use the virus clinically, it may then, we may get an opportunity to ask those questions in patients that have failed all other kinds of therapy. But right now, this is our strategy for this virus. Yeah.
This is more or less linked to this question. Don't you think that the cancer cells would behave as a population?
Absolutely.
More or less like, for example, the European rabbits in Australia were infected with the virus, like
and we get asked this question a lot. What about escape? Escape mutants. The truth is, escape from therapy is what cancer cells do. And it's true for chemo, it's true for radiation, it's true for all therapies that exist right now. And so what oncologists do is they look for layered ways to get rid of every last cell. So there's no one magic bullet. Childhood leukemia today is mostly curable. And it's curable by sequential use of various drugs that oncologists learned how to use one after another. The drugs were invented in the 50s. There's a wonderful book out there called The Emperor of All Maladies. It's written by Siddhartha Mukherjee. If any of you are interested in cancer research, pull up the book. It might even be translated in Portuguese now. But if not, it's a wonderful read. And it describes how scientists and clinicians and oncologists went from having powerful drugs that by themselves just killed patients and learned how to use them clinically. And they went from childhood mortalities of most leukemias, the mortality in the 50s was 90%. By the mid-1970s, the same drugs, and learning how to use them, they went to like 5% mortality. They went to a curable pediatric cancer. And a lot of us in oncolytic virotherapy feel the same. It's not a magic bullet, but it will be a tool to help clear out certain aspects of cancer cells that will form part of a strategy in the future. That's my answer. It's not a single magic bullet because of that exact fact. You've got to get every last cancer cell, and you have to have multiple weapons to be able to do it. And we hope this will be a weapon in the future. We'll see. Maybe your kids will know. Yeah.
Does it work without a rabbit Poxvirus?
Don't know. We did a couple of experiments trying to purge myeloid leukemia with a couple of vaccinia versions. We were using vaccine versions. We tried MVA and we tried LT16M8, which is a vaccine strain from Japan. And the reason we picked those is because they're both licensed for use in humans. But neither purged the myeloid leukemias very well, not as well as myxoma. So we sort of dropped it. And we've never really gone back and looked at it because our plate is full just with myxoma. So I would say it has not been properly explored yet with other poxviruses to be able to give you a really good answer. Any other questions? Okay, thanks.
Thank you.